Okay, hi everyone. So thanks for joining my talk. So this lecture is a part of our Fundamentals of Fluorescence Microscopy series. And today I'm going to give you an introduction to bioimage analysis. Some of you that I can recognize name. My name is Ellie Cho, and I am an application specialist for image analysis at Biological Optical Microscopy platform at the University of Melbourne. So if you have any questions, feel free to type your questions in Q&A and I will answer them at the end. And also the recordings will be available on our website afterwards. So from now on, I'm just going to turn off my video. I'd like to start by acknowledging that the University of Melbourne recognizes the traditional owners and custodians of the lands on which we work and learn and their contribution to our community. And also, I'd like to introduce the Light Microscopy Australia, or LMA. So LMA is a special interest group of the Australian Microscopy Microanalysis Society. So as a member, you get access to travel bursaries, data analysis workshops, and also regular events updates. One of the big events that's coming up is Australian Conference on Microscopy and Microanalysis in Sydney next year. It is the 4-day symposiums with 2 days of workshops. Also incorporate LMA and Bioimage Analysis group called AEST. And you can join at microscopy.org.au or follow us on LinkedIn. So in this talk, I will cover 6 stages of image analysis workflow, starting from planning, acquisitions, pre-processing, object detection, measurements, and reporting. So along the way, I will pause for a few quick pop quiz questions to check our understanding together. So let's start with the first stage, which is planning. Let's have a look at these 2 fluorescence images. So the left is a control group, and the right has been chemically treated. And when we want to measure the differences, we might describe maybe the cell treated looks a bit rounder, but this needs to be backed up with proper quantification. And one useful measure is a circularity, where value of 1 means a perfect circle, and closer to 0 means it's more elongated. And once we measure circularity for each cell, we can then plot the values and run statistics. So this is pretty much the goal of bioimage analysis, to obtain quantifiable information from microscopy images of biological samples. But the thing is that image analysis actually sits at the end of the experimental workflow, because we still need to prepare the sample and acquire the image first. And also, as microscopy and image analysis techniques becomes more sophisticated, there's much more to consider. And the most common mistake is neglecting this complexity, which then leads to incorrect analysis or having to repeat the entire experiment. So we need good planning to avoid this. So the first step is to determine what needs to be measured, then determine the microscope techniques that will generate image that's optimal for that measurement. Then prepare your sample accordingly. And this type of planning must happen before any data collection begins. But sometimes, because the biology may be unknown or the expected observation may differ from reality, so we always need to run a pilot study before committing to a full-scale experiment. And then use the pilot to optimize settings, readjust plans, and determine your final sample size. And once optimized, then we run full-scale experiments. And if you're not sure where to start, I guess the best way is consult your local core facility, or you can contact us early in your process. So let me introduce the general workflow for bioimage analysis. Starting from raw microscopy images, the general workflow usually involves pre-processing to correct any acquisition defects and segmentation to detect the objects or define a region of interest, then measurements of the relevant parameters. The final is reporting the result. Also, depending on the analysis type, we may use a single software package covering the whole workflow or combine multiple tools for specific functions. In terms of the tools and softwares, there are many processing softwares available. So if you look at here, there are over 1,400 tools that are curated on Bioimage Informatics Index at BIII.eu, which is developed by the Network of European Bioimage Analysts. So this is helpful for finding specific tools, though it can be really overwhelming if you are new to this field. There are a couple of useful review papers, but for most people starting out, I recommend Fiji, which is built on ImageJ, which is the most widely used open source platforms, where it also gives you access to hundreds of plugins. So let's start with the first step of image analysis workflow, which is acquiring your images. So acquiring good images is critical for good measurements. And since the microscopy basics are already covered in our previous lectures, I'll focus on 5 key factors affecting the quantifications, which are resolutions, sampling rate, bit depth, dynamic range, and saturation. And to understand the acquisition requirements, let's first look at how the microscopy image is generated. So the fluorescence signal from your sample is formed into a continuous analog image through the microscope optic. And when it's detected by the camera and saved to the computer, this signal is then converted into digital format, which is pretty much a grid of pixels. And each pixel has a defined size and also the intensity value. So the first step in controlling this image in terms of controlling the image resolutions is first choosing the correct objective lens. If we were to use the wrong lens, means then analog image won't have sufficient resolution to start with, neither the resulting digital image. And the objective lens selections and how it determines resolutions is already covered in our first lecture, so if you have missed it, please check it out. And the second factor is the sampling rate, which is used when converting the analog signal to a digital image. And to do this correctly, we need to follow the Nyquist sampling theorem. So the Nyquist sampling theorem, in case you aren't familiar with it, it's pretty much saying that to accurately represent a signal digitally, you must sample it at least twice the highest frequency that in the signal, which sounds a bit complicated. So what does that actually mean? Let's have a look at this diagram. Say the black curve is our signal, and the red dot is where we will be sampling. So if we sample 1 point per cycle, this is pretty much the result, which doesn't really represent the real signal. And increasing the sampling rates improves the result. And when we sample at 2 times of the signal frequency, it starts to reflect the signal shape bit properly. So this is the best minimum requirements that we need to use. In microscopy, this translates that your pixel size should be at least 2.3 times smaller than the smallest object you want to resolve. So for example, to resolve a feature about 10 microns in diameter, your pixel size should be no larger than 4.3 microns, which is a factor of 2.3 rather than 2, because it actually accounts for optical diffractions as well as the square shape of the pixel. So here you can see the effects of the different sampling rates on the same object. If we undersample it, which means using the pixels that are too large, it results in a pixelated image where all the fine details are lost. And when we oversample it, it generates a sharper image, but at the same time, it takes a lot longer to acquire, it produces larger files, and also slow down the entire processing. So really the goal is a sufficient pixel resolutions without under or oversampling. Okay, next is the bit depth. So every pixel in your image has an intensity value, and when we convert the signal to a digital image, we can simply convert as 0 and 1, or into 4 gradient steps, or 8 steps, or 16 steps, on and on. So these images are encoded as 1, 2, 3, 4 bit depth, where you can kind of see the bit depth essentially determines how many levels that intensity can take. So an 8-bit image has 256 levels of gray values, which looks smooth enough for human eye, and that's why it was historically the standard type. But for quantifications, usually the higher bit depth, like 12 bits, that gives about over 4,000 levels, or 16 bits, that gives over 65,000 levels, that records intensity difference much more precisely. So as an example, in visually, here between 8-bit and 16-bit image, visually you won't see the difference, but the quantification will be significantly more accurate. So we always want to use higher bit depth than 8-bit for intensity measurement. The next is the dynamic range and saturations. So the intensity distribution of an image is shown in a frequency histogram, like you can see on the right. And ideally, the histogram should be using the full available range, like this. But if the image is taken too dark, the signal may not separate from the background, which then gives poor quantification. On the other hand, if it's too bright, pixel can become saturated, which means that they've reached or exceeded the detector maximum. And once the image is saturated, we can no longer know the true intensity value within those regions, and the quantification become invalid. So we always want to check the histogram and using the saturation indicator during acquisitions and adjust the settings accordingly. And when we acquire a fluorescence image, each fluorophore is captured from a separate spectral band, which we call this as a channel. So in this example, you have 3-channel image, channel 1 to 3, that was acquired at 12 bits. So each pixel has an intensity between 0 to 4095. And in microscope software, each channel is stored as a monochrome grayscale image, like this. And then in specialist software like Fiji, we can apply a color lookup table, which is called LUT, to display the channel in the color of the fluorophore as close as possible. But note that this is only a display settings and the underlying 12-bit data stays intact. But the problem arises when we try to share or publish images. Because the standard image viewer don't support LUT display, the images often get converted to RGB type. So the RGB image type stores the color as a 3 separate channels, red, green, and blue, but each at only 8 bits, which means it's compressing the information from 4096 down to just 256 intensity level. And then when we try to splitting the RGB type back to the individual channels, the image with RGB LUT that was becomes reduced bit depth. And also, if a non-RGB LUT was used, such as yellow or cyan, then the information is completely corrupted and cannot be recovered. So the take-home message is always keep your original monochrome raw files and measure from those, and don't measure from any RGB exported image. Additionally, a few important points to keep in mind. The first is any images you plan to compare must be acquired and processed with identical settings, like differences in laser power, detector gain, or exposure time that will invalidate the comparison. Second, always save the raw file formats from your microscope, such as CZI, LIF, ND2, OIR, or even OMETIF, because these formats preserve the 2 critical types of metadata. The first is the calibration factor, where the pixel size, Z spacing, and time interval. And without this calibration factor, your measurements are just pixel counts with no physical unit. So there won't be no micrometers, no area, no speed measurement. The second is the acquisition settings like laser, detector, object magnifications, which are essential for your records and also for publishing. And also when you save it, standard formats like TIFF, JPEG, PNG strips most of this information out. So only export to those for final figures, but never measure from them. That's the key takeaway. Okay, it's just me talking the whole time. So let's test if we remember some of the important factors of acquisitions. So what I'm going to do is I'm just going to launch a quick quiz. Hopefully you can see 3 questions. They're pretty simple of repeating what I've just mentioned. And I'll give you maybe 1 or 2 minutes. Okay, so I'll just stop the poll and share the result. Can you all see the result? And the responses? Hopefully you can. And I will, good, thank you. And I will reveal the answer here. Ooh, that's good. I think most of people have selected the right answers. So let me just quickly go through this. I think I might have gone a bit too fast with this Nyquist sampling theorem, but essentially what you want to remember is this value 2 times. So as long as you make your pixel size about 2.3 times smaller than your object, then you will be safe in resolving your structures. And for the second questions, in terms of the bit depth that you want to use for intensity measurements, I'll say anything higher than 8-bit depth. So in this case, I put it as a 16-bit, but also if your camera only has up to 12 bits, that's still fine, still a lot better than 8-bit. So please have that in mind. And the last one is which format is most appropriate for saving microscopy images for intensity quantifications? Yes, most of people have selected the right answer, which is a raw file format. And so in Fiji and many other specialist softwares that deals with microscope images, you should be able to open these raw file formats and access all the data fine. The standard TIFF, as I mentioned, that's still a lot of people using it, but be careful because it may actually have removed all of your metadata. So thank you very much, and I'll just stop sharing. And I'll move to the next, which is pre-processing. So even when you have followed all the acquisition rules that I mentioned, raw microscopy images can still be compromised. So in many cases, we still need to correct them before analysis. So there are many procedures available. In this talk, I will cover the 6 most common, which are denoising, background corrections, bleaching corrections, alignment, Z-projections, and deconvolution. So each microscopy image contains noise, and they are mainly coming from electronics of the detection system, and this noise can interfere with object detection. So the common options to reduce noise is using image filters such as the median or Gaussian filter. The median filter replaces each pixel intensity with the median value of its surrounding pixels. So it's fairly effective in reducing the noise, but also at the same time, it preserves the edge information very well. The Gaussian filter applies the Gaussian convolutions mainly to smooth or blur the signal. So it's slightly different between the median and Gaussian filter. But also more recently, there are a lot of deep learning approaches have been developed for denoising, such as the tool named CARE is a type of supervised method which means you need a paired noisy and cleaned image for training. Noise to void, N2V, on the other hand, is unsupervised method where it pretty much estimates and removes the noise from the noisy image itself. So there is no clean image pair required. Also, the uneven background illumination is common where you can see the center of the image often appears brighter than the edges. And that's mainly due to the optics and misalignments within the microscope. And the common tool to use to remove this is rolling ball background subtractions. So in this method, it estimates and subtracts the background based on the user-defined radius. But if there is more complex unevenness happening, we can also use the flat field image, which is an empty background image that's taken with exactly same settings. And then we can use to correct by dividing the raw image using this flat field image. Sometimes there is a stripe artifacts, which mainly comes from the camera system. That can be corrected using a bandpass filter. And for more advanced corrections, tools like Basic can compute and correct shadings from the data itself. So these tools are available in Fiji, ImageJ, and Self Profiler, and many other programs. Next is bleaching corrections. So in fluorescence imaging, so intensity can decrease over time or across the Z-stack, mainly due to the photo bleaching of the fluorophore or the fluctuation of the light source. And this can make quantitative analysis difficult. So this type of bleaching can often be corrected by fitting an exponential decay curve to the data sets. As you can see, it can be corrected. But sometimes the decay, if the decay is not exponential, we can also try different method called histogram matching, as you can see in this example. And usually the Fiji bleach correction plugin or bleaching corrector in Huygens program handles things like this. And when we do this, we have to be careful and use these bleaching corrections only for segmentations, but we shouldn't really use this for intensity quantifications because it actually alters the actual intensity values. Another type of artifacts is the image misalignment. So this type of misalignment can occur in multidimensional data and mainly due to the sample drift or stage movements over time. So this must be corrected before detection or any measurement. But also the misalignment can occur between the fluorescence channels due to the chromatic aberrations, which means the different wavelengths focus at slightly different positions. So this is especially important to correct before any colocalization measurement. So tools like Transform J in Fiji and the chromatic aberration corrector in Huygens can help with this type of misalignment. And when images are captured at multiple positions to form a mosaic, then they must be stitched into a complete image first. And big stitchers in Fiji can be a good, powerful tools for this, but there are many other stitching programs available as well. I guess most of you probably have dealt with the 3D images. So when working with the Z-stack, we often collapse them into a single 2D image before analysis. And when we do this, the type of projection we do matters. So I guess the most common one is the maximum intensity projections, where pretty much it picks the brightest pixel at each XY positions across all slices. So this is useful for sparse and bright structures, but the object at different Z depths can appear to overlap. So we have to be careful with this. The sum intensity projections adds all pixel values across Z, making which means it makes more appropriate for total intensity quantifications. But at the same time, the backgrounds also accumulate too, so we have to be conscious of this. And the other type of projection is average intensity projections, which means it averages information across the slices. So this is good in terms of reducing the noise, but it can also dilute signal from sparse structures. So, but in the end, if your objects are densely packed around the Z volumes and none of these projection may not appropriate, and in that case, 3D segmentation is the better approach. Then lastly, the deconvolutions. So the deconvolution is the mathematical image restoration method, and based on a model of how light scatters in your microscope, which is called point spread functions. It tried to restore the signal to increase the resolutions in XYZ and improve contrast and remove noise. So as you can see in this example, the localizations and the separation of object in green and red objects will be much more accurate after deconvolution. So there are commercial softwares like Huygens, and it's available at our platform. So feel free to contact us if you like to access. But there are also a couple of other free tools if you want to go for that option. Okay. So this is the end of the second part. So I'm just going to launch where my quiz gone. Sorry. Oh. Okay. I'm just going to launch the second one, which is only one questions. So hopefully this should be straightforward. Right, let me share the result. I think most of people have selected the right answer, which is good. So the question was, which features can be improved during pre-processing step? So where I've shown some of the image filters as well as the deconvolution method, it can improve the signal to noise ratio. Unfortunately, it cannot recover the saturated pixels. So once the pixel been saturated, as I mentioned in the previous section, the information is completely gone. So that's why we have to be really careful in acquiring the right image to start with. The third answer is relatively easy. So if deconvolved, it will increase the resolutions, yes. And also I've given you a couple of examples of how we can correct the background illuminations. And in terms of the increasing bit depth, you can say if your images were taken at 8 bits and then you try to increase to 16 bit, it's not going to increase or add more details. It's just going to change your image into 16 bit, but the information will either stretch accordingly or just stay exactly in the same intensity value. So there's no gain in gaining. We're not getting any more information after through the pre-processing step. So it also has to be set during the acquisition properly. Okay, just stop share and let's move on to the next step, which is the detecting the objects that we want to measure. So I guess probably the simplest approach of the object detection is a local maxima detections, where we try to find the brightest point in each region. So this is useful for quick counting, but it gives no information about the size or the shape. So for more details, we use segmentations, which means we're pretty much classifying the pixels as signal or background and converting them into a binary image. And after segmentations, we can do additional processings like splitting touching objects or grouping the pixels into individual particles. So I will first cover these three main segmentation approach in the next slide. So in conventional segmentations, what we do is we set an intensity threshold to separate the signal from the background based on the image histogram, like this. And then this results in a binary image, which is black and white. When we use a different threshold value, obviously it's going to give a different result. So we need to set this objectively. And the simplest approach is to use a negative control image with no expected signal. Then we can set the threshold based on that image and then apply the same threshold to all the rest of images for consistency. But in real life, the background levels often vary between images, so making a single fixed threshold ineffective. And this is where adaptive methods are needed, which is called auto-threshold method. So these methods calculate the threshold value logically within each image. And there are many algorithms available. For example, the method called isodata tries to find the threshold where the average intensity of pixels above and below the threshold are equal. The method called intermode assumes there's a histogram with a bimodal shape and is trying to find the midpoint between two peaks, in this case M1 and M2, and there's a middle point there. And because different methods work better for different image type, it's always important to test several methods and choose the most appropriate. So in Fiji, for example, you can test 17 different auto-threshold methods and 9 different auto-local threshold methods in a single step. And you can browse through the method, and once identified, then you can apply the same algorithm consistently across all images in the dataset. So which means the actual value applied to each image will be different, but it will be calculated based on image histogram, but using the same type of mathematical algorithm, which is still considered more objective rather than we try to find the value visually. But still, this type of conventional thresholding struggles with low contrast or non-fluorescence images such as bright field, phase contrast, color image, or electron microscopy images. So in this case, machine learning can help. So in machine learning, user annotates a small number of pixels manually using something like a brush tools. And this is typically quick and very interactive. And then software analyzes multiple pixel features like intensity, texture, color, and local context. And it calculates the probability of each pixel belonging to each class and generates a labeled image like this. So there are many free tools available, including the trainable Weka and LabKit in Fiji, Elastic, CubePath. So most of these are user-friendly interface and also require no programming. But there's also deep learning models that can detect much more complex patterns in challenging images. So for example, like UNet can segment individual cells from bright field or EM images, which is really low contrast like this. Another deep learning model called StarDist is really good at detecting the round object like nuclei, inflorescence, and both histology. And another really popular deep learning model is Cellpose, which is a very versatile tool that handles various cell shapes. The downside is most approaches require large amount of manually annotated training data and sometimes the programming skills. But there are also a lot of pre-trained models available. So for example, like in Bioimage Model Zoo, they create this type of models. And also many can be accessed directly through Fiji, Elastic, Coupa, Cell Profiler without any coding experience. So once images is segmented in any of these methods, like conventional, machine learning, and deep learning, then what we get is this type of binary image. And this binary image can be refined with morphological operations. So for example, the watershed splits these signals and separates touching particles. Distance map assigns the pixels with increasing intensity values from the object border. So this can be used to identify or filter objects by their proximity to other structures. Boolean logic operations allow filtering based on overlap with the other channel. So I will demonstrate some of this in the workflow examples coming up. But before this, let's have a quick break and check our understandings on object segmentations. So I'm just going to launch our third quiz. It has 2 questions. Okay, I'm going to stop now. And the share result. Right, so the first question was in conventional segmentations, what is the best way to set threshold on dataset with various background levels? And yes, majority of people have selected the right answer. So rather than applying a single threshold value, which can then may not be applicable for different images with different background levels, we can calculate the background level for each image, but using the same type of algorithm. So the second answer, applying the same auto threshold algorithm, will be the right answer. For the second one, for which image type is conventional intensity thresholding least appropriate? Yeah, definitely. Bright field, like color image, EM image, those in real life, when I see those images, I'll probably just go straight to the machine learning or deep learning method and don't even bother using the conventional intensity threshold because the intensities of my signal to backgrounds are much different. Okay, so I'm just going to stop sharing and move to the next part, which is the measurement stage, which is applying quantifiers to the objects that we've detected. So there are many different type of measurements, and the most common 6 are number, intensity, shape, distance, colocalizations, and tracking. So I'll walk through each of with an example workflow. Let's start with the number. I guess one of the most common workflow is counting a positive object. So in this example, we have 2 channels, one showing the nuclei on the top and one showing the phosphorylated histone H3PH3, which is the marker for mitotic cells. And let's say we want to count the percentage of cells that's in mitosis. The first step would be detecting individual objects in each channel, so nuclei area and mitotic cell area. And very often we want to remove anything that's touching the image border, as those are usually the incomplete objects. And then using some Boolean logics I mentioned, we can filter the nuclei based on whether they overlap with PH3 object. And we score 1 if they do and 0 if they don't. Then this gives us a total count and positive counts, which then can be plotted or shown as a result overlay. And another one is intensity. So to compare the brightness between images, obviously the mean intensity is the right measure. It is a sum of all pixel values but divided by the object area, which gives the size normalized measure of staining level. So in this example, we want to measure how much of protein has translocated from the cytoplasm to the nucleus. So this is another typical intensity measurement. And first, because we have the nucleus staining and the whole cell staining, we can detect each region and also subtract the nucleus from the entire cell, making the cytoplasm mask. And so once we define the nucleus and cytoplasm area of each cell, then we can use the protein channel to measure the intensity. And then we can calculate the nucleus to cytoplasm mean intensity ratio as an indicator of translocation. So these are really common ones. But beyond the mean intensity, we can also measure minimum, maximum, median, sum, standard deviations of intensity. And as an example, these two cells have similar mean intensity. So just measuring the average intensity and checking the brightness won't really differentiate their patterns because you can see the right one is clearly more granular. But when you look at the histogram of each image, the intensity distribution is somewhat wider on the right image. So in this case, we can measure the coefficient of variation, which is a standard deviation divided by the mean intensity. And this can capture the dispersions of the histogram and also can serve as a measure of fragmentations or heterogeneity of the intensity. Also, the biological structure can be measured by many shape parameters. So, for example, these two cells, they're very different looking, but when we measure the simple area, they are very similar. So it doesn't really distinguish their character very well. Whereas if we measure the ferrite diameter, which is the longest line that can be drawn across the object, reveals that the right cell is more spread. So that's a good one. We can also measure the parameters, which measures the total surface length. But in this case, those two cells have also very similar parameters. We can also measure the convexity, which is the ratio of the object parameter to its convex hull parameter. So the convex hull is the shape that's formed by stretching, let's say, a rubber bands around the object. You can see the yellow outline shown here. And it's measuring the ratio of those two parameters. So in this case, the right cells has lower convexity values, indicates it's more irregular and also concave surface. So this type of different shape measurements can be possible. But obviously there's a lot more and more, and the full list of shape measurements you can find from ImageJ documentations or the shape filter plugin, which can be very useful. Also, the distance between objects is a very common spatial measurement. The nearest neighbor distance measures the centroid to the centroid distance between the objects. So it's computationally simple and also works well when the features are small and relative to the actual distance between them. So when they're very well separated and the objects are small, then you'd measure this nearest neighbor distance, and also when objects are round. But when objects are large compared to their separations like this, and also irregular in shapes, or their edge-to-edge minimum separation distance is, I guess, more important, then you can measure this border-to-border distance rather than the centroid. So here's an example workflow where we want to identify a synaptic particles that are physically touching a specific cell. So how do we do this? Is we can first generate a distance map outside the cells. As I mentioned briefly, the distance map is an image where the pixel intensity increases gradually from the cell border outward. Then for each detected particle, the synapse particle, we can measure the minimum distance map intensity. So if we measure the intensity of the distance map, a minimum of 0 means the particle is actually touching the cell border. And then we can filter the particles to keep only those within the minimum intensity of 0. So these are the touching objects, pretty much. The colocalization analysis. This is really popular analysis, but at the same time, it can be easily misinterpreted. So we have to be very careful. So in bioimage analysis, colocalizations indicates that two fluorophores are generally near each other. So it does not mean the molecules are on the same locations or they are interacting. So we can measure the correlation between channel intensity using a Pearson's coefficient. So it's more asking whether if the green is brighter, red is brighter. So it's looking at those correlations. Or we can also measure the co-occurrence using Mander's coefficients. So it's looking at each pixels and whether those two pixels overlap or not. Or we can also measure the object-based overlap, where it will, so it doesn't have to be a pixel by pixel, but it's more object by object. And also other very important factor is this colocalization measurement is highly dependent on the image resolutions. So as you can see in this image, the image taken with low resolutions, you will pretty much see high colocalizations artificially because they're all blurred out. So we have to be really careful in planning the right acquisition settings if we are going to do the colocalization measurement. If you're working with a time-lapse image, we can track the specific parameters like speed, displacement, accumulated distance, direction, or straightness, and the durations. For example, here, the objects from different conditions were tracked, and their displacements, as in how much they moved, has been plotted for control and the treated cells, which you can visually see the reductions in movement after treatment. So this is what you can do in the tracking. Before we move to the reporting parts, there are a few important points that I like to highlight on what we want to do with the measurements. The first question is, we want to understand what your correct N is. So let's say you have measured 500 cells from a single animal or single experiment. Your N is 1 rather than 500. So these 500 cells are technical replicates. So so it reflects the precision rather than biological variability. So for biological conclusions, you need independent biological replicates, like separate animals, cultures, or experiments. I think this is what a lot of people get confused. That's why I decided to put it here. And the second is we want to visualize the data appropriately. So if you look at the top, the bar chart, which is the very standard format, that hides the distributions of your sample. And more people emphasized either using the swarm plots or violin plots to show every data point that reveals the spread, skewness, and outliers. And finally, we have to be aware that many image measurements like intensity, shape, distance are not normally distributed, which means that before applying simple t-test or ANOVA, first check your distributions, and if the data is skewed, use non-parametric tests like Mann-Whitney or Kruskal-Wallis. So ideally, all of these considerations should be planned before you start to collect any data. Okay, so let's check our understanding on the measurements. Okay, I think most people have answered. So let me just end the poll and share the result. Okay, I think looks like message has been passed through relatively successfully. So the first question was what's the best quantifier for comparing brightness of microscopy images? Yeah, the answer is absolutely a mean intensity. The coefficient of variations came a bit high. I guess the coefficient of variation is really measuring how varied your intensity is within the object. So if you want to measure the granularity or heterogeneity, that might be a better measurement. For the second one, the select correct description of colocalization analysis. So it's a multiple answers, and yes, it doesn't measure the interaction of two molecules. If you want to measure the actual physical interactions, it's better to use different type of microscope techniques like FRET. But in image analysis space, when people are referring to colocalizations, it measures the correlations of pixel intensity like Pearson's, or it measures the general proximity of molecules like Menders and object overlap. And obviously, yes, I've shown you that image taken with low resolutions will inherently have the high colocalization regardless. So if you're planning to do it, try to take image as high resolution as possible. So yeah, I think most people will select the right answer. So I'm just gonna stop sharing. And then this gives us to the last stage, which is reporting and documenting your analysis. So this is one of the published examples that shows the analysis workflow. As you can see, it has like a nice flowchart with example images. It's clearly documented as in this way. So, but also shows how complicated the image analysis workflow can be, right? So some softwares like Cell Profiler makes this relatively straightforward. So you can build a pipeline by adding or removing individual modules, and the entire workflow is saved as a file, which then can be shared or reused, which is excellent for reproducibility. So if you want to learn more about this workflow and also other examples, there are many example templates to help you how different workflows are structured. So please visit their websites here. And for programs like Fiji and QPath, which are other two popular open source programs, they don't really have a built-in pipeline environment. But instead, the workflows can be recorded through the script recorder and replayed for batch analysis. There are other scripting options, includes like IC, Pythons with scikit-image, Napari, MATLAB, and KNIME, and more. So these scripts can be also a very important part of your publication method. But at the same time, not every biologist needs to become a programmer. So if you need help in automating your analysis, you can contact your local image analyst. So for people based in Australia, you can contact us Bias Group or reach out to our platform. And in terms of reporting the measurements, it's common to skip workflow details. So then that becomes a reproducibility issue. You probably have seen people saying just image was analyzed in Fiji in their paper. So this vague descriptions is not really sufficient. It's never going to make anyone else to replicate that work. So the bioimage analysis community has published a reporting checklist. It was published in Nature Method to address this. So when publishing a new workflow, what we should do is we should clearly describe each component and the sequence of the steps, and also upload your workflow and sample data to an online repository like GitHub, and also explain the rationale and limitation of the approach. And ideally, we can also include the screen recordings or tutorials. So the checklist at least is work as a guideline indicating what needs to be published when you are, what needs to be added when you're reporting your work. But also there is a checklist that covers the best practice for publishing microscopy images. So it's really worth reading before any image publication. So the last section was relatively short. Let's quickly check our understandings on our reporting. So I'm just going to launch a last quiz. Yeah, I think most people have answered, and I'm really happy to say that everyone got the right answer. Woohoo! So yes, just to reiterate, when we are reporting our analysis, we need to report individual components used. So components in this case, I would say type of software and type of plugins. And if it's in the big software, what type of module was used. And obviously, if you include the parameters, that would be even better. And if it's completely new workflow, then it's always good to have the example data set so that people can actually try and understand what can happen. And none of the image analysis workflow will be perfect. So it's always good to mention what's the limitations as well as why, what sort of rationale was used within there. So I guess everyone would appreciate if someone actually published their workflow this way, then you can actually replicate the work properly. Okay. So I'm just going to put to the next slide, which is the last page. So to show there are some online resources here. So as I mentioned, our website hosts materials from previous workshops, including this series, but also the video sessions. So I recommend you check back regularly, especially if you're learning. And also the Image SC forum is always my first stop for finding tools, troubleshoots, and workflow recommendations. It's really active community with all the excellent discussions all around the world. So this is really important page to bookmark. And also each software has its own websites with manual tutorials and work examples, which are really great for learning for specifics. So thank you very much for attending, and I'm going to hang around to answer your questions. So feel free to type any questions in the Q&A, and I'll try to address accordingly.